if I struggle with this English language, it's because I've only spoken it for the last 6 years. If you don't follow me, just put up your hand and let me repeat the word. I'll try several versions of that, and hopefully we can follow each other eventually.
If they follow me, they can follow you.
Yeah, I guess that's true.
Being British.
So the talk this morning is really about the GlycoFi technology, what we have done and where did it come from. And I might need to just... This is just sort of a quick overview of that. I won't spend too much time on that. What you can see on there, hopefully from the back, is basically a crystal structure of a monoclonal antibody. It's of the type IgG, which basically means that it's got a heavy chain and another heavy chain and then 2 light chains. The top end of the monoclonal antibody, that's sort of the business end. That end is responsible for binding the antigen. If you look at this in 3D, it looks like a pair of hands that's sort of opening up and grabbing its target. The bottom end of the antibody really interfaces with receptors. And I'll leave it at that for now. We're going to come back to that a little bit later. In this pocket between the 2 heavy chains, there is drawn sugar structures. Now, for those of you who do not know what glycosylation is, but glycosylation is basically the attachment of sugar moieties or sugar structures onto a protein backbone. So these sugars are covalently attached to asparaginins inside that pocket. Obviously they give structure to that pocket, obviously they form and they shape that pocket, and that is important for ADCC, which is antigen-dependent cell cytotoxicity, complement activation, macrophage binding, distribution, and clearance. So the glycosylation, although in the case of antibodies is hidden inside this pocket, plays a very important role in the functioning of the antibody. Here's another protein. This is probably the most contested biotech space in the world at the moment. The white, blue, whatever color you want to call the structure is the erythropoietin receptor. Just quick recap, erythropoietin stimulates the formation of red blood cells. So if you plan to run the Tour de France, it seems to be in fashion to take a little bit of erythropoietin to boost the red blood cell count so that the availability of oxygen is much more, and therefore you can do much better. The erythropoietin itself is sort of that part of the picture. Now, erythropoietin has got 3 end-linked glycosylation sites. And the sugars that are attached to that are shown over there, and over there. In the case of erythropoietin, obviously this erythropoietin has been made in mammalian cells, in fact in CHO cells. And what you see here is different samples and basically an in vivo activity. These 2 samples were made in roller bottles. These ones were made in a fed batch reactor. Those ones were made in hollow fiber reactors. So depending on how you produce this erythropoietin, there is a 500-fold difference in vivo activity. Pretty scary stuff. Why? If you analyze these samples, what you guys probably cannot see, but guarantee to you, the difference between these samples sits in the glycosylation only. All of them have the exact same amino acid backbone. Same protein, same sequence, same folding. The only difference is the degree of sialic acid. Sialic acid, being a sugar moiety, happens to be the very last sugar moiety on that. So the challenge that the industry face with the production in mammalian cells is that if you secrete a protein in mammalian cells, you don't get one glycoform out. You actually get a whole mixture of glycoforms that come out of these mammalian cells. And each of these glycoforms has got a different activity or binding site or has got a different function. There is a structure-function relationship between glycosylation and how that occurs and how the molecule functions. So here you can see this is basically an assay that looks at glycoforms, or the structures of the sugar moieties that are being attached. These happen to be the structure that for erythropoietin gives you the longest half-life. The problem with erythropoietin, it's a pretty small molecule. If it's not highly glycosylated, what happens is you inject it, it spends a little bit of time in your body, and then it goes out quite quickly through a filtration mechanism. So what they have found is that if you want that erythropoietin to hang around in your body for longer, what you've got to do is you've got to enrich for a glycoform that is sort of expanded and it's got lots of sialic acids on that. That makes the hydrated volume of the molecule much bigger. That prevents clearance. So the picture here is that, look, the CHO cells produce very little erythropoietin. Of that very little, only 10% is part of the sample that you want. So our friends at Amgen, who obviously dominates this market, have to throw away almost 90% of the erythropoietin that they make because of the inherent problem that CHO cells has, which is it produces a heterogeneous mixture of glycoforms. All right, so this is where GlycoFi came in. Traditionally, we had human cells. There's some gene that we want to express. Let's, for example, use erythropoietin. We put that downstream of a promoter that we know the strength of. We put that into a CHO cell, and we make it. And as you can see, the color of this is slightly different than the color of that, because the protein is very, very similar. It's got human glycoforms, but it's not exactly the same as the one that's in the human body. If you want to express this in yeast, your problem is now the color is very, very different. Yeast cells traditionally do have the ability to glycosylate proteins. Now, let's back up a little bit. If you think about E. coli and all other prokaryotes does not have the ability, well, mostly does not have the ability to glycosylate any proteins. So if your protein that you want to express is glycosylated, you have to use an eukaryotic expression system. If you use a mammalian eukaryotic expression system, what you get is all human glycans, but you get a mixture of glycans, which for the best part is the highest sort of status of the technology. And that was quite acceptable. You can inject that stuff into humans, they don't die, it's not immunogenic because those glycoforms are all present in the human body. So that's quite okay. But you know that CHO cells grow kind of slow. It takes 14 days or 21 days for a process. They are very shear sensitive, so you can't shake them too hard or stir them too hard. You can't even look at them too much, and they break. So it's a pretty sensitive system. Yeast, on the other hand, let's face it, people have been making wine and bread and beer for many, many, many years. Right? These buggers are pretty robust. You can shake them, you can stir them, you can throw them around. The other thing is you can make a lot of them. CHO cells, they need fancy media compositions, all sorts of serum-derived things. Whatever you do in the CHO cell business, it's just dollars. Yeast cells, look, they can grow on salt and water. These buggers are pretty happy growing on anything. So there's a lot of cost advantages. The downside of the yeast system is that the glycosylation type or the sugar moieties that they attach to the proteins are not human, and they're not even closely human. In fact, it is believed that they will trigger an immune reaction, that they are actually immunogenic. So if you make a protein in yeast today and you inject humans with that, there is a very high likelihood that you'll have an immune reaction to that, which for most patients that is already deprived of something is kind of, unwanted. So that was the big challenge. What GlycoFi then did is they said, okay, well, fair enough. If we can change this yeast cell, rip out the plumbing, basically prevent this yeast strain from glycosylating proteins the way yeast cells do, re-engineer all the human glycosylation pathways, we have the best of both worlds. We have a system that is robust, inherently cheap, easy to scale, easy to use in any bioreactor anywhere in the world, that can also then make human proteins. So that was sort of the GlycoFi mission. Now, to do that, you need to understand a little bit about how proteins are made and how proteins move through the secretory pathway. So what we've got here is sort of a cartoon of the secretory pathway. Protein production starts in the nucleus. The protein gets translated, moves out into the ER. From the ER, it moves into the Golgi, into a cis, a medial, a trans-Golgi, then goes to a trans-Golgi network and makes its way outside of the cell. So I think the picture that I'm trying to paint here is this is like an assembly line. That guy, Henry Ford, that came up with the assembly line, that's the image that you've got to have here. This is a very sequential process. The protein travels along this road. All right. How does glycosylation work? Human glycosylation, this is a sort of an image of a protein. Just use a little bit of imagination. The protein moves into the ER. What is being attached is this core oligosaccharide. Then there's a glucosidase that takes off those glucoses. Then you get this structure. Then there's a manosidase that rips off that structure. And this is sort of the glycan structure that will move into the cis Golgi. All right. Now, what happens in yeast cells? Guess what? Exactly the same. All right. This was good news very early on. This basically means that the early processing steps through the ER for yeast cells and for mammalian cells are conserved. And we did not have to do anything there. All right. Next step. Now we move into the early Golgi. There's a manosidase that takes away those mannoses. Another manosidase that, first the glucnac that adds on that glucnac is a glucnac transferase. A manosidase that takes off the other two manosidases. Another glucnac acts and add a glucnac. Moves to the late Golgi. We add galactose. We add sialic acid. And that is sort of the glycan structure of most human proteins. What happens in yeast cells or in fungal systems? Remember, it's the very same structure that enters into the early Golgi. First thing that happens in yeast cells, there's an addition of a mannose right there. If you can add one, you can add many. All right? If you can add them in this direction, you can also add them in that direction. So there's a whole array of mannoses that's being added, and that gives you these high mannose structures. This structure is very, very different from that structure. This structure is human. This is fine. If you have that on a protein, your body would not recognize that as a foreign body. This is non-human, and it's most likely immunogenic. So what did we do? The first thing was we need to get rid of that first addition step. If you can prevent this first addition step, those steps can't happen and these steps can't happen. That step is triggered by an enzyme called OCH1. So what we did is we found the gene, we deleted the gene, and if you delete that gene, what you do is you prevent this addition step. Right? Now we got the yeast cell back to step one of the human cell. We basically got this structure back by making that deletion. Okay, so that deletion happens, that addition happens somewhere in the very, very early Golgi. Now the next step is that, because you'll remember that this is a sequential process, it's like an assembly line that the things move through, what we need to do is we need to go and line up these enzymes one by one in the assembly line, because each of these enzymes basically need a substrate from the previous set. Think of the assembly line that Henry Ford built. The chassis of the car's coming in, you've got to put the wheels on, then you've got to put the lid on, the engine in, and if you don't do that in the right order, the whole thing doesn't work. The yeast cell works exactly the same way. You've got to go and add these human enzymes, and you've got to make sure that they act in a very specific sequence, because each of them depends on having the substrate available. So that basically means that we need to go and put a manosidase somewhere there. We need to go and put GNT1 and manosidase 2 somewhere over there. The other GNTs and the GAL transfer needs to go there. Sialic acid needs to happen over there. Now, how do we do that? Most of these glycosylation enzymes are type 2 membrane proteins. Now, what does that mean? A type 2 membrane protein has got a catalytic domain and it's got a stem region. The catalytic domain is responsible for the action. So a manosidase catalytic domain is responsible for removing or cleaving a manosidase. The stem region is sort of responsible for localization. That stem region is specific to a specific spot in the secretory pathway. So the problem was that we don't have catalytic domains or stem regions that we know where they're going to locate. People have predicted that, and they more or less had an idea where these things would go, but nobody knew that for sure. So what we then did is we said, okay, we're going to make leader sequences. We're basically going to take enzymes, whether those be human, frogs, flies, dogs, rats, anything that is eukaryotic, that has got leader sequences that we know, well, we don't even have to know where they localize. As long as we can sequence and clone the leader sequence, we're going to make a library of that. At the same time, we're going to make a library of a whole array of catalytic domains. See, what is the pH in the secretory pathway of the yeast cells? We don't know that. How are you going to find it out? There's no way to measure that. All right? So how do you know that the catalytic domain from a human monocytase 1 is going to work inside a yeast cell? Because the physiological conditions may be very, very different. The fact is you don't know that. And nobody's going to know that. So what we did is we said, well, we're going to clone catalytic domains from fungus, from worms, from frogs, from flies, from dogs, from rats, from plants, from any possible thing that has got a catalytic domain. And then we end up having two libraries. We've got a library of catalytic domains, and we've got a library of leader sequences. So what we do is we have our leader sequence library, we have our catalytic domain library, we make a set of plasmids of all possible combinations, we put that into a single plasmid, we transform that plasmid into a yeast cell, that you get colonies back, and now all we need is a readout. So our readout is glycosylation structures. So what we did is we picked these colonies, we grow them up in a 96-well plate. We use a robot to purify that because obviously you've got hundreds and hundreds of colonies that you want to look at, and you don't want people to do that all by themselves. They get purified. After they get purified, we load them on a mass spec, and then by mass spec, we just look at these colonies one by one. Mostly what we got is this structure, which is the man-8 structure, which basically means that the catalytic domain and leader sequence did not work. This is the exact structure that we started off with. So pretty unexciting. And you get actually quite a few of those. Every so often, you see this peak, which basically means now you have a pair that did produce the desired effect. If you look hard enough and you look long enough, you can actually find something that's pretty pure. Now, mostly you have this single glycoform. In fact, 95% of the glycans that is produced by this cell line is of this form. At that point in time, we realized this is already very, very different from the CHO cell business. You remember when I showed you the picture of erythropoietin? Many, 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 many peaks. Interesting peak, quite a small fraction. If you look at this picture, almost only one peak. 95% of the total glycans is one structure, and that happens to be the structure that you want. All right, unfortunately, that's not all we had to do. We also had to go and engineer in monocytase 2, the GNT1, GNT2, and for all of these, we just followed the same process. So you pick the winners from the first round, then you take sort of the new library, new catalytic domain, fuse them together, screen them, and look at them. For GNT, just as an example, it was not that simple either, because glucnec as a substrate did not exist in the ER of yeast cells. Yeast cells don't need that. In fact, it's in the ER, they need it very early on, but then in the gluc, it's not there. So even if you have the right catalytic domain and the right leader sequence at the right point, you don't have the substrate. So it's not going to work. Right? So what we had to do is we need to build a pathway to get the glucnec out of the ER and move it into the right spot so that that is available at that point in time. To do the galactose addition. Galactose doesn't exist in our yeast strain. It's not there. All right? They don't want to consume the galactose either. So what do you do now? We basically had to convert glucose using an epimerase inside the cell to galactose. Then made sure that that galactose is UDP linked, and that is in the right spot in the Golgi, that you have the right leader sequence, the right catalytic domain, and the substrate all together so that you can get this glycan structure. Yeah. And all of this is really achieved by combinatorial libraries. Many, many libraries, many screening. The success in this really lied in the ability to clone fast and efficiently and to screen fast and efficiently. That's sort of the novelty of this. All right, this actually only gets you halfway there. Then we had to go and add a sialic acid. Now, sialic acid, once again, doesn't exist. The substrates for that doesn't exist. You basically had to go and build an entire new pathway to make sialic acid inside the yeast cell. Then once you've made it, you've got to get it into the right spot, right catalytic domain, right leader sequence, and only then the action will happen. After, 5 years of work, this is what we published. You basically have a starting strain that is what we call a GS 5.0 structure, meaning that it ends in terminal galactose. We can then prove that we can get sialic acid transfer, and then just to prove that this really was sialic acid, we mixed in sialidase, which is an enzyme that cleaves off sialic acid, and then it goes back to the original picture. So to take a wild-type yeast strain out of a box and to convert that into a strain that can glycosylate proteins in the exact same way that the human cell would do took us at least 14 genes that we had to clone in. And it's not just simple clone it in and it'll work. It's cloning it in at the right spot, at the right place, in the right order, so that the whole pathway as a whole could work out. So what do we have now? These are all sorts of glycoforms that you can find in the human body. All right? To date, nobody really knows what these glycoforms can do. Why not? Because they don't have ways and means to make that. Out of a CHO cell, you can get all of them, but you get them as a mixture. How can you separate them? That technology is very, very difficult with very, very low yields. And to be honest, it's not 100% efficient. Now we have a library. And you can basically say, well, which one of these glycoforms work the best? And which one do I want? Because we've got strains, if you want that glycoform, we'll make that glycoform at this fidelity. As you can see in the picture, there's one peak, one glycoform only. You know what? We can make that one, and we can make this one, and we can make that one. And, I mean, you can really go to town. It just keeps on going. Obviously, all of these things are published, and this was tremendous work done by the geneticist at GlycoFi. What can you do with this technology? Why is this useful? I'm back at human IgGs. The reason why I'm talking about IgGs or antibodies is really that's probably the biggest growing segment of the pharmaceutical industry. I think today there is 28 approved, fully intact antibodies, and I'm not sure whether that's a 2006 number, and give or take, that's more or less where it is. If you look at the pipeline of the big pharmaceutical companies, every big pharmaceutical company has got several antibodies in their pipeline. If you look at the predictions, this is the space to go. These are wonderful molecules. Just quick reminder, IgG1 has got a heavy chain, it's got a light chain. Glycosylation happens somewhere there. Glycosylation important for various things. Here's an interesting story. If you look at antibodies that are being produced by mammalian cell culture, so in all the lane 1s, what you're going to see is the mammalian cell culture product. In the lane 2s, you're going to see the GlycoFi product. So the first one is an isoelectric focusing gel. What this gel does is it looks for the pI, basically the pH at which the molecule would be neutral. What you can see for the mammalian cell is 1, 2, 3, 4 bands with a little bit of a smear in between. The reason why you see that is the mammalian antibody that's being produced consists out of an array of glycoforms. These glycoforms give the molecule a different charge. If you look at this just by SDS page, with or without a reducing agent, so this is without the reducing agent, so there you see the fully intact antibody. That's with the reducing agent, so there you see the heavy and the light chain. They look pretty much the same thing. If you look at this by reverse phase or size exclusion, same thing. If you look at the glycans, you can see that the GlycoFi yeast, predominantly a single glycoform, the mammalian cell culture, a whole array of glycoforms. Why is that important? Let's talk a little bit about a molecule that is, well, how do I put this? ADCC, and the example that I want to use is Rituxan. This is a molecule that binds to CD20. Now, it is being used at the moment for treating non-Hodgkin's lymphoma. So what happens is you have these tumor cells. In this case, it's a B cell. On the surface of that, there's a CD20 receptor. Then antibodies will float around. The business end of the antibody, or the binding end of the antibody, would recognize the CD20 on the surface, and it'll bind to that. The bottom end of the antibody, which is sort of the FC region, that is responsible for recruiting the natural killer cells. The natural killer cell then sort of does its thing, kill the cancer cell, which is quite nice. And the story is that an antibody has got two binding sites that are both equally important. It's got to recognize the antigen, but then it's also got to have the ability to recruit the NK cell so that the NK cell can go and do its work. All right? That interaction happens through an Fc gamma R3A receptor that's sitting on the NK cell. That interaction is critical for ADCC to work. So if you look at Rituxan at the moment, there's two polymorphisms in human beings with respect to the receptor. Right? About 60% of human beings have a valine at position 158. If those people happen to get non-Hodgkin's lymphoma, the Rituxan would work for them. The rest of us basically have a phenylalanine at that spot. The phenylalanine in that spot basically means that Rituxan is not going to work for you. What you can see on the graph is the depletion of B cells. Remember, those are the bad boys. We want as many of them to die as quickly as possible. If you take commercial Rituxan out of a vial with this phenotype, you can see that the depletion is actually very, very poor. If you were to make this molecule in a wild-type yeast cell, where you have these high nanostructures, what happens is you don't get it at all. It doesn't work. It's completely useless. If you make this glycoform, you can see now suddenly you have depletion of B cells. If you can make this glycoform, now that goes even higher. So this was clear proof that glycosylation plays an essential role for antibody binding to its receptors. And it's not only the presence of glycosylation, it's having the right glycans there. Now, if you make this molecule in a mammalian cell, and only this glycoform is the one that does the binding, let's assume you need a gram of antibody to treat a patient. Only 10% of that is the right glycoform. That means you need to inject 10 grams to get the same effect. Whereas if your entire gram had this glycosylation pattern, that means only 1 gram would be sufficient. So this is sort of where the GlycoFi technology is going, is we would start, well, I guess Merck would like to look at what is the relationship between glycosylation and the function and the structure of therapeutic proteins, including monoclonal antibodies, and then start to exploit that further and further. So I guess this is almost straightforward. We believe that GlycoFi's technology is going to change the way in which in which therapeutic proteins will be made in the future. And at this point, I'd just like to sort of acknowledge everybody in the GlycoFi team. And the list is very, very long. And all of these people were obviously key in achieving this to date. That's it.
Two-part question. First part is with reference to, I believe it's Candida elegans or Cryptococcus.
Which is that?
The species, the yeast species.
That we used?
Yeah, Candida elegans.
No, we've used Pichia pastoris.
Yeah, Pichia, yeah. But I also saw C. elegans.
Okay.
Yeah.
The C. elegans is basically where we got catalytic domains from.
Okay. So,
so it's not that we used C. elegans as a production system. That was basically just where we got catalytic domains from.
okay. The second question is that long time ago I studied lipoproteins from Rhodotorula, which seem to produce a lot of lipids. And while screening for the catalytic part of the moiety, the reaction moiety, have you screened a lot of other genera of yeast, probably the Baryomyces, just as a screening process?
Fungal.
Yeah. Mainly I'm concerned with the yeast part of the fungal domain. So have you screened any of the other species of yeast to see where you can maximize your yield, like Rhodotorula or Torula or Cryptococcus?
What is that, as an expression system or as a source of?
Expression system as well as a source, so that we can zero in on the one that can produce more.
Well, we've worked on several yeast systems, so our technology is broadly patented in fungal systems.
Okay.
So glycosylation in any fungal system is something we've proved, and we've proven that technology in more than one system. In terms of what would you choose as an expression host, now I think there are two major schools of thought. There are people that believe that you've got to have the right system. So it's important to look at as many systems as possible and to find the one system that works well. Another group of thinking says that, look, it doesn't really matter what system you pick, as long as you can understand how to adapt that system to maximize your own approach. I'm not sure that we have fallen in any of these two categories. I think both has got pros and cons. In our case, we've sort of chosen Pichia pastoris to move forward with that, because taking this technology forward with 50 type yeast strains really doesn't make sense. If you can take it forward in one yeast strain and that strain works for you, then why would you stop? The cloning technology in Pichia is well known. The genome in Pichia was well known. How to clone Pichia, I mean, there's a kit on the market that you can go and buy that'll tell you how to do that. So from a molecular point of view, look, the challenge to get this done was big enough. We didn't want to go and figure out how to clone in the species as well. As an expression system, Pichia is well published. I mean, people make 10 grams per liter of HSA in that. If you look at FC fragments being secreted in Pichia and you look at the literature on that, it's gram per liter quantities. So the ability of Pichia pastoris to secrete proteins is well proven and well established. And the fact that the FDA is regarding that generally as safe, sort of a bonus. That helps quite as well.
Very impressive.
I actually have a couple of questions. One is the leader and catalytic domains. The ones that work, were they mainly from other fungal species, or were they all across, or can you not say?
No, I can't say. It's a very good question. I'm smiling because it's a really good question. We plotted them, and we try and predict. And the truth is, it's chaos. There is no way of predicting. We looked at that data, and we represented it in most of our publications. It's like a color chart. We have, X and a Y axis of combinations and the colors, and you can move these any way you want. There is really no pattern. Okay. So I think what I'm trying to say is that you would not get, previously, what was different about GlycoFi? People knew about glycosylation many years before. Various other very clever groups have tried this approach before. Their approach has always been, I'm going to take this leader sequence and this catalytic domain, and those two are going to work. What we said is that is unlikely to work, and we were right. There was no way that you could predict that. You basically had to have the library of both, screen them, and if you can do that efficiently, you basically you don't need to understand why they work, because they do work, and that was the novelty. So in our production strains, what is common? It's a spread, anything from mammalian to human, and all in between.
And the second question, do the genes that you've inserted, are they stable? I mean, the metabolic, the growth of the cells and the stability of those constructs?
Very, very good question. So that's another reason for using Pichia pastoris, is we integrate these genes directly onto the chromosome. They go into the genome. We integrate them in a way that will make them to be stable. It's a double crossover event. Okay. There's no repeating sequences. We've tested these strains for as many generations as we think they will ever see. And to date, we found all our constructs to be stable. We have found ones that are not stable. And then we can trace it back to, look, this is the way it's been integrated, or that's sort of the case. But the strains that we have now is stable for longer than what they'll be needed to produce whatever the protein is that they need to produce. It's true that if you do engineering, you do end up getting different growth phenotypes back. But the fortunate thing on our end is that, having set ourselves up as a high throughput screening facility, we really have the ability to go and find the clones that has got the attributes that we like. So what we've done is, yes, you do a transformation, you get clones back that, they're pretty sick and, they don't look too happy. But if you look hard enough, you actually find clones that are as happy as the wild type strain. They grow fine, they produce fine. The titers at which we produce is obviously proprietary. I will not be able to disclose that. What I can tell you is that we are very competitive with CHO cells. The CHO cell industry has been around for a little over 10 years now. When they started off making monoclonal antibodies, the titers were 2 or 3 milligrams per liter. It took them 10 years. They are at about, let's say, 5 grams per liter at the moment. We've been at this for a little over than 2 years now. Initially, we didn't even... Making a monoclonal antibody in a yeast strain is not trivial. People have not attempted that. They've made FC fragments and fusions and things like that, but making a fully assembled functional antibody in a yeast strain, that by itself was quite a challenge. This is a complicated tetramer that needs to fold exactly right for this thing to work. So we've sort of taken that road on 2 or 3 years ago, and we are already on par with CHO cells. I'm predicting that 5 years from now, we're going to look back and say, well, where are you? I get paid to say that, by the way.
That's related to Tom's question. One of your strain, I guess, GF16, you said there were about 14 gene insertion in there. Sounds like it's a lot of gene insertion. What kind of selection method do you use? And it's kind of strange. You don't have any human homologue of one of those 14 genes you inserted there.
We don't have human homologs?
Mm-hmm.
Well, of those 14 genes, a big portion of them are actually human genes.
I mean, the yeast homologue, I'm sorry.
Yeast homologs?
Yes.
Well, on a DNA level, that homology is very, very low. Look, glycosylation pathway doesn't exist in yeast. So to expect that a yeast strain has got a silidase transferase in it is not there. They don't have the need for that. Okay. So therefore, a silidase transferase is a novel and a new gene with virtually no homology to any other piece of DNA that's inside the cell. The fact that you need 14 genes to do this, I think that is a lot. And I think that's what really makes this technology impressive, is you can literally look at this, that we took the yeast strain, we ripped out the plumbing, and we rebuilt in a whole new road. And whatever that took to do that. What's even more impressive is that this new road, or the yeast cell, perceives this road as if it was there forever and ever. It doesn't perceive this as being a foreign object. It doesn't want to get rid of it. It's quite happy to live with that. In fact, as you move down the pathway, these yeast cells become happy. They really have no growth phenotype. And I think, could we predict that when we started? Not a chance. When we initially applied for money, people laughed at us. They said, you guys from where want to do what? This will never work. It wasn't obvious that this is the road to go by. We know that the cell wall of yeast cells depend highly on the glycosylation structure. So even we predicted that once you get beyond a certain point, there will just be no cell wall integrity, and you'll be back at the mammalian cell system. So the fact that it turned out quite opposite was kind of exciting, and makes this technology, well, I guess, really worthwhile. I think Merck made a very good investment. The CHO cell product will basically have an array of glycoforms. Remember, for erythropoietin, depending on how you make it, the glycoform is actually very, very different. So if they made their phase 1 batch in a roller bottle, but they want to do their actual production in an airlift reactor, the FDA is going to say, but no, you can't do that. In our case, independent of how you produce this and where you produce this, the yeast strain produces a very well-defined glycosylation pattern. And the fidelity of that is very, very high. So the uniformity from a glycosylation point of view that we can bring to the market is actually higher than what the current trend in the market can offer. We actually believe that the FDA would look at glycosylation characterization quite differently once we start to bring things to the market. And they're going to say, well, look, if these guys in yeast cells can do that, you CHO people need to step up to the plate, and you need to sort of demonstrate the same uniformity. That's the one angle to that, where I think we have an advantage, where I think the FDA would like to see that. The other angle is fair enough. This is a new production system. There are proteins made by Pichia pastoris on the market and sold today, so there is some precedent to get this through the system, but it is a new production system. And I do expect that they would require of us to prove safety and non-immunogenicity quite extensively for the first molecule. Once you've established that precedent, we'll be back in the same shoes as where the Cho people are at the moment.
Have you only used this technology for this one product, or have you looked into it for multiple products?
We've looked into that for several products. I cannot tell you how many genes we've expressed, but it's probably more than 50, if it's not more than that. So we've looked at monoclonal antibodies, we've looked at therapeutic proteins, we've looked at everything that is being expressed on the market, novel proteins. In our early days, we've had collaborations with big pharmaceutical companies where they would bring their favorite protein to us. We would express it in our system, they would analyze it, and we would get some feedback. So I think our system, at least in our own hands, have been fairly broadly tested. And the future will tell how much of that is applicable and how much of that we can really push through the pipeline. The best I can say now is, watch the space. It's going to get very interesting.
And use protein A for purification. Is that true? I mean, like everybody else does for the
antibodies.
MABs. Yeah.
Yes.
Yes.
I mean, the nice thing is the protein is secreted, so it's outside of the cells. You don't have to bust open the cells and deal with all of the proteins that's inside the cells. You remove the cells by centrifugation or filtration or whatever your method of choice is. You take the soup, and then that soup looks the same as what the CHO cell soup looks like. It's no different.
Except it doesn't have it doesn't have
no viruses, no serum, nothing to deal with from that point of view. So that soup is inherently safer than the soup that you get off the CHO cell system. The total protein content that you need to compete with is obviously significantly lower, since this is a secreted protein. So even from a purification point of view, there's very definite and defined advantages in using this.
So when you're talking about that, are you trying to focus a lot on animal-derived component-free in your entire process?
Oh yeah, without a doubt. Yeah, our current media composition has got no animal-derived components. In fact, our facility is animal-derived component-free, so that the strains that we want to move forward is not contaminated by animal-derived products. And it's really because the FDA frowns on that, and something we don't need, so we don't need to compete in that space. Why? I'll tell you afterwards. Well, I mean, I think publicly there's a very, very good synergy between the two companies. Merck wants to get into biologics. They're quite serious about biologics. Merck took our technology very seriously. They treated us very, very well. Our relationship with them to date has been extremely positive. It is tremendous how much effort they've made, to make us feel welcome in this big company. Remember, we were a company of about 60 people living up in New Hampshire in the woods, doing very cool science, but not interfacing with the real world much. Now we're part of a massive corporation. The whole way in which they handled the transition process has been really, really neat. The synergies from a business exploratory point of view in terms of where they want to go and what we can offer and what we can bring made a lot of sense. It's been a very, very fruitful partnership.
I wanted to comment on the people, because I know that you have more countries represented. Could you just say something about your
Sure. I think if you look at the list, it's really spot the American. We have people from every continent in the world. It is a very, very diverse group of people. I think in the very early days we were only 12 employees, of which I think 2 of them were American, or at least born in America. So it's very diverse. It's people from all over the world. It's people that Tillman Gerngross and Stefan Witt recruited really based on their ability to contribute to the science. So it's a lovely environment to work in. I mean, there's no better colleagues than these guys. If I look back at the past 5 or 6 years, it's been great. It's, you know, we've done top-notch science. We've done that in a place in this world that's as beautiful as it gets, and we've done that with lovely people. It's really been a privilege. I mean, I think it's a neat experience, and it's a pity that most people will never experience that.